Okay, thanks for joining everyone. I see 11:02 Pacific time, so we will kick off today's webinar. This is the first in a RareCyte 2021 webinar series, and this webinar is entitled Circulating Tumor Cells, Delivering on the Promise of Liquid Biopsy. I'm Leighton Howells. I'm Senior VP of Biopharma Partnerships at RareCyte, which means I'm really focused on working with our clients on the development of custom assays and clinical sample testing to meet their clinical trial needs. With me today is Arturo Ramirez, who's our Director of Oncology R&D, who leads our amazing R&D team in the development of those assays and the running of that testing. So thanks for joining. We will be taking questions principally at the end of the presentation, but if we see any that pop up during presentation, we'll try to answer them as we move along. So thanks again for joining. So RareCyte is a Seattle-based life science company with about a 10-year history in the commercialization of high-performance imaging and precision engineering tools. We really have a vision to provide the next generation of tools for both liquid and tissue biopsy, tools that are really enabling for both research and clinical studies. Today, we're only going to focus on liquid biopsy, but in a future webinar and maybe in the fall season, we'll talk about Orion, which is our latest technology for highly multiplex tissue imaging, up to 21 biomarkers on a single tissue slice. But for today, we're really going to talk about liquid biopsy, and we're going to get into a number of things. Really, we're going to describe a little bit about why liquid biopsy has such a potential. Arturo will give the bulk of the presentation, talking about assay development and assay validation. He'll look at multi-omic data analysis and show you a range of case studies on dual biomarker assays on CTCs. And then finally, we'll give you a sneak peek into some emerging data from a couple of key collaborators showing the prognostic value of CTC counts in epithelial cancers. So, lots of people say, why liquid biopsy? And really the value here is that it truly enables therapy selection and real-time patient monitoring in an ongoing fashion. In the US, there's about 2 million cancer cases per year. Globally, it's about 10 times that. And while significant ground is being made in immuno-oncology, it still is typically only about 30% effective. So there's a lot more that we can do. And to do that, we need to have routine testing available to patients. Classically, IHC and tissue biopsy is the standard of care, provides great information about the tumor and about limited numbers of biomarkers, but really is a one and done scenario. It's very difficult to get repeat biopsies from patients. In things like lung cancer, there's up to 25% adverse event scenarios. So, from a liquid biopsy perspective, it's minimally invasive. It's both safe and cost effective compared to tissue biopsies. And that means it can be done at greater frequency, means potential greater access to patients to life-saving drugs. And really importantly, both protein and molecular data is available from a liquid biopsy, certainly from circulating tumor cells, that can support cancer research and clinical applications. At RareCyte, we really modeled our high-level workflow on the tissue biopsy as the standard of care, so that our technology platform can slide easily into an histology lab type environment. So what that means is there's a sample collection, there's processing of the sample to slides, and then there is imaging and analysis, where the imaging piece of it provides great insight into phenotypic information. When you think about liquid biopsy more broadly, there's a range of analytes that are possible. Cell-free DNA, very commonly used for mutational studies. CTCs, as we'll discuss today. Exosomes emerging that we don't have listed here. But when you compare the liquid to the tissue, what you see in this chart is that, yes, you can get molecular information from both these analytes, but you cannot get protein biomarker information from cell-free DNA. And really the power of an intact cell like a CTC is to be able to directly quantitate and understand localization of that biomarker in an intact, potentially live cell. So that means that you can get heterogeneity information that you cannot get from a cell-free sample. And also it means, of course, that you can monitor, like you can with cell-free DNA, that you cannot do with tissue. Later in the presentation, Arturo will show you a study of monitoring a triple-negative breast cancer patient for 50 weeks, taking a blood sample every week. And so the volume of data and the monitoring of those patients is enabled via this approach. So coming to our platform, we really have created one platform for discovery through diagnostics. The platform that has the flexibility for discovery and research, but also has a robustness and performance and infrastructure to support diagnostics. As you'll understand as we go through the discussion here, our platform is end-to-end from sample to result. The flexibility enables multiple biomarkers to be looked at on a single cell and look at those markers and change them as needed. But also flexible breakpoints in the process where you can simply bank samples for staining and processing later. You can bank samples while the assay is being developed, for example, while you're generating batches at CROs for processing. So a lot of flexibility in the process. And then really the last aspect on the left-hand side here is that this is a comprehensive assay scenario. So I talked about, cell-free DNA being complementary. From the processing of a RareCyte sample, we also extract a high-grade plasma that you can then use for cell-free DNA, as well as the bulk CTC, as well as the ability to pick individual CTCs for molecular assays. So you have really sort of three different analytes from one scenario here. On the right-hand side then, this is more around the infrastructure. Arturo's team develop and can validate assays to clinical specifications. We can deploy them in a client lab with the appropriate instrumentation. We can deploy them at CROs or quality service providers. We can transfer to those service providers. And then we can also run everything from pilot assays through to companion diagnostic programs within our services lab. This takes us to the sample to result workflow, and it really is a simple process to deploy in any lab. It is something, as I mentioned, that really aligns to sort of a standard workflow in a histology lab, where we're collecting a blood sample, 7.5 ml blood draw. We're using our processing device to transfer that blood to a slide where processing is unbiased in its isolation of nucleated cells. So we collect all the cells and all of the rare cells. We don't select for them based on size, shape, morphology, biomarker. We transfer what is called the entire buffy coat to 8 slides, and then we stain all 8 of those slides. We image them using high-performance imaging that takes around 10 minutes, and then we report and review. And the way that the process runs is that we have machine learning algorithms that will provide a rank score for what is likely or unlikely to be a CTC. And these algorithms were trained based on thousands of clinical samples and now many, many years of use, where it's very clear that they identify the CTCs with a very high rate. But then for final check, we have trained reviewers just confirm any marginal decisions that the algorithm will have made. So that is the typical workflow. Takes less than a day to run end to end. In more of a research phase, we can then use one of our instrument systems, actually the top one that you see on this chart here called a site finder, where that site finder can pick individual cancer cells from that buffy coat, process it to a PCR tube, and then that can be processed for further molecular analysis, such as DNA sequencing. So really a very simple process and a very efficient workflow to isolate with high reproducibility, incredibly high sensitivity, all of the nucleated cells and the rare cell population within. So zooming out a little bit, this is now how the assay would be deployed in a more of a clinical trial workflow. So where the blood would be collected at a clinical site, It would be placed in a shipping box, and then from that shipping box, it would be transferred to a processing lab. We call our sample prep Exquisite. It's that little device that you see in the middle there, unbiased in its collection of CTCs. Samples are processed to slides. They can then be stored. They could be run at that location, or they could then be transferred to another lab for testing. And really the breakpoints here are that there's 72 hours of blood sample stability for the shipment. We have data that shows it can go 96 and beyond, but for maximum collection, we're recommending 72 hours. And that's usually enough on a geographical basis to ship slides to the processing site. Once processed, we then have, I would call this at least 12 months of slide stability. Arturo's team are currently showing 2 to 3 years for CTC stability, and a similar sort of sign for a large number of biomarkers that we've looked at on those CTCs, showing that the vast majority of them are stable over multi-year storage. So again, that means that trials can be run, samples can be stored, and analysis can be performed at a later date. We're often asked about what indications shed CTCs and how many do you get? And so this is a short list of indications that we've looked at. We'll show you others later on, where we found CTCs. The numbers range from anything from single digits to tens to thousands of CTCs per 7.5 mils of whole blood. The value that's brought here is that our recovery is incredibly high. So if there's a single CTC in a 7.5 mil blood draw, the RareCyte process will find that CTC. And clearly it's well understood that CTCs are prognostic in prostate, breast, and colon cancers as an indication of progression, of patient survival progression. So mostly what we would say here is CTCs will be found in all indications in varying numbers depending on the patient themselves, the stage of the disease. But what is really has to be well understood is that CTCs have long been known as the source of metastatic disease. They are the vehicle in which cancer unfortunately moves around the body. And metastatic disease, of course, is one of the key challenges, perhaps the major challenge in cancer treatment. So getting to a cancer cell, understanding its phenotype is critically important. On this page, we're showing you examples of a range of CTCs in a number of indications. What you can see when you look at the panel on the left-hand side, they vary in size, they vary in shape, they vary in whether they're isolated or in clusters, and they certainly vary by biomarker expression and the localization of that biomarker. So the imaging data here really gives a lot of value, not only in terms of count and biomarker quantitation, but where that biomarker is within that cell. And on the top right, we show, again, a range of indications where we've developed assays and looked at CTCs, and then a short list of biomarkers that we've created assays for. These are the ones that are publicly sort of visible. We have a much longer list of client confidential biomarkers that are being, either have been developed or being developed within our biopharma programs. So coming to that assay development, this is a very sort of simple workflow, again, of how we approach it. We have a milestone-based process for the development of those assays. It obviously starts with customer requirements, and that's mainly about what is the biomarker, what are the indications of interest, what is the grade of assay that the client might be looking for. Is it an exploratory mode pilot assay, or do they want to use this in RUO mode in more sort of retrospective trials, or do they actually want to move into more of an RUO companion diagnostic mode? And so here we're really just focusing on the pilot as the starting point for all of those assays, where once the client requirements are understood, we then move to design and optimize that assay. It's a lot about identifying the appropriate antibodies. And what we would say is, an antibody that works in IHC is very likely to work in a CTC assay. It may not be the best antibody. And so in that case, we will screen at a wide range and determine which are the best antibodies. We obviously then need controls, and so identifying positive and negative control cell lines, deciding which autostainer can be used. We work very closely with Leica and Ventana. Both autostainers work great for this process. Sometimes one versus another might be better for a particular assay. And a lot of that has to do with sort of how the cells are fixed and antigen retrieval and then stained on the scanner. So again, there's an optimization phase before we move to the validation phase, which is really about determining the performance of the assay. And we'll do that both on model cancer cells, but then also on clinical samples, either commercial samples or maybe some of the very earliest trial samples. And we'll validate that assay in terms of its performance. Arturo will show you a lot more of the data, but really what we're trying to do is create an assay where, you know, the vast majority, 95% or more of the CTCs in a blood sample will be found, trying to do that reproducibly, and then also ensuring that biomarker information meets sort of our expectation in terms of how it aligns to controls. So, you know, a process that is milestone based, typically for these types of assays, might be a 12-week process, obviously longer for more highly validated assays. But once we've gone through that design phase and optimization phase, that's pretty much the assay ready to go for validation. So with that as sort of a brief introduction to liquid biopsy and the platform, I'm going to pass to Arturo, who will take you through a good amount more data and start to get into a discussion about the types of biomarkers that we've looked at and the types of performance that we've achieved. So with that, over to you, Arturo.
Thank you. Let me just share my screen and my video. Good morning, everybody. I'm going to talk to you today about assay development and validation process at RareCyte. So one of the main benefits of our platform is that it's flexible for the user's particular research goals. And one example of this flexibility is that there's 2 extra biomarker channels available that could be added to the CTC detection assay. So the way we find CTCs, we typically refer to it as the base assay, and it has 3 channels that allow us to find the very rare CTCs amongst all the white blood cells on that slide. So those 3 channels would be the nucleus, CD45 for human blood, and then CK and EpCAM to find epithelial cancer cells. And in addition to these 3 markers, there's extra 2 biomarker channels that you can add any biomarker of interest here. You can use any combination of mouse or rabbit primary antibodies. So you could have 2 mouse antibodies, 2 rabbit antibodies, or 1 of each. And this developer technology is the same technology that we use at RareCyte to design new assays for our customers. So as an example, I'm showing here at the bottom a lung cancer assay that was done. And you can see the 2 extra channels we chose are vimentin for epithelial mesenchymal transition and the epidermal growth factor receptor protein. So as you can tell here, the vimentin is present on all the white blood cells, as well as this spikin A549 cell that we added to the blood. However, the EGFR is only on the CTC of interest. Another example of the flexibility of our platform is the cell discovery kit. And this allows the substitution of the human CD45 that I was mentioning earlier with mouse CD45, so you can detect CTCs in mouse samples, such as PDX samples. Also, you can change the CK-EpCAM biomarkers to a non-epithelial biomarker of interest to find non-epithelial rare cells in your blood of interest. And in this case, I wanted to show a assay that we built for melanoma testing. In this case, it's for human melanoma samples because we kept the CD45 anti-human antibody there, and we substituted the cytokeratin and EpCAM antibodies for an S100 antibody to detect these rare melanoma cells. And so now you can use this on melanoma samples. Besides the flexibility of our system, in order to find very rare cells, sensitivity and accuracy are paramount. So to quantify these two metrics, on the left, I'm showing the sensitivity of our assay, where what we did is we took 9 tubes of blood and spiked in very low numbers of CTCs with a very precise instrument that counts these cells. And then we took them through a whole process from blood all the way to result. A blinded reviewer did all the reviewing. And at the end, you can see that we got 96% recovery, even at these very, very low numbers of spike in cells. So extremely sensitive assay at finding these rare cells. And then for as far as the accuracy, on the right-hand side here, I'm showing a graph where we tested 50 cancer patient samples against the FDA-approved cell search assay. And this was done in a laboratory at the University of Washington. And they actually certified our assay in their CLIA lab to run this test. And they did the blinded study where 2 tubes were taken from the same patient at the same time. 1 tube was taken through the RareCyte platform. 1 was used with the cell search technology. And as you can see here, there's extremely high correlation between both of these assays with a very high R-squared value. However, the RareCyte platform had about 1/3 more CTCs on average than the cell search assay. We've done a little bit of investigation on this, and the reason is we think that because the cell search assay requires both cytokeratin and EpCAM to be expressed on the same cell at the same time to detect the CTCs, our platform only requires one or the other one. In fact, if they're both at very low levels, there's a little bit of a synergistic effect where they're both in the same channel, and so we can detect very low amounts of CK and EpCAM on the CTCs and therefore have a higher degree of sensitivity than cell search. So besides the accuracy of detection of the CTCs, another important characteristic of our platform is that we're very accurate at detecting the biomarker of interest, and we can quantify these biomarkers. And so here I'm taking you through a little bit of what we do during the validation of a new assay, specifically for a novel biomarker, where we select cell lines that are positive and negative for the marker. And then we set a threshold that best separates the positive cell line from the negative cell line. And then with this, we can quantify the level of sensitivity, specificity, and accuracy to determine biomarker positivity. So for this first example, I'm showing a PD-L1 assay that we made. And for this, the positive cell line is the SW900 cell, and the negative control is the HAP1 cell line. And because we get single cell mean fluorescence intensity, and it's automatically generated by the system, we can either determine biomarker positivity as a simple binary yes or no, but you can also have single cell biomarker expression. So each one of these dots here on these dot plots is a single CTC that was found on a cell, so it's fully quantitative. And you can see here for this assay, we have excellent accuracy, sensitivity, and specificity. Another performance metric that we quantify on our analyses is slide-to-slide precision. Basically, it's a measure of variability that is seen between different slides on the same run or on separate runs. So in this example, I'm showing a different assay, which is the synaptophysin assay. And it shows a coefficient of variation that ranges from 6 to 16.1 within the same day. So that's the repeatability. And then on different days, the CV is just under 14%. So what I'm showing here is each one of these is a slide. Each slide has about 100 model CTCs, and there's 5 replicate slides on each of 3 different days. And so this is the 10th and 90th percentile, and then this is the interquartile range. So you can see the variability within a day and then between days, and we quantify it with our assays, and we want to make sure typically it's under 20% so that the assay is robust at finding the biomarker of interest. Another key aspect of our platform is you don't only get the single cell intensity measurements as mean fluorescence intensity of the extra biomarkers, but you also see high definition images of where that protein is expressed on the cell of interest. And this can be important for certain biomarkers where the localization of the protein is important for its function. So on the left, I'm just showing again a dot plot of a ER versus HER2 on the same cancer cell, and you can see the 4 quadrants similar to a flow plot. And then on the right, something you don't get by flow, which is the single cell morphology and biomarker localization for the single CTCs. Next, I'm going to talk about the downstream genomic analysis that can be performed on single CTCs that allows us to characterize these to a much deeper level. So single cell genomic analysis is made possible through the site picker module, and it's installed in some of the modules of our site finder instruments that they're the automated slide scanners. And the workflow for isolating single cells is very automated. It involves first you scan the slide as normal, up to 5 channels, then you remove the cover slip. Then because the scan already knows, the instrument already knows where the cell of interest is on the slide, you just introduce the slide back into the instrument without the cover slip. And this very small needle comes down and picks that single cell of interest off of the slide. And this is an automated procedure that happens. And the cell of interest then goes into that needle and then is deposited into these flat bottom PCR tubes. And we make these ourselves, and they have a flat bottom. So then when the needle basically goes into the PCR tube and deposits the cell of interest here at the bottom of the tube, the instrument is imaging in real time. You can see the needle come down and deposit that little droplet, 50 nanoliters of fluid, with your cell of interest there. And then from here, the PCR tube can then be taken to do downstream molecular analysis. The whole process from picking the single cell off of the slide and depositing into the PCR tube takes about 3 minutes and is highly automated. So single cell next generation sequencing is something that's performed routinely in our lab. And I want to show you 2 particular interesting examples of the findings that we've made with this application, both in a study of triple negative breast cancer, and it was performed with Tony Blau at the University of Washington. I worked on this study personally in the lab a few years back, and I was in close contact with the physicians involved in the trial. So what I'm showing in the top left is data from a first patient that was followed, and Leighton commented on this data earlier. The patient was followed for more than 9 months, and we took 53 blood samples approximately once every week, and we analyzed them for CTCs. So her the CTC counts of this patient came in extremely high at 1,500 per milliliter of blood. Upon treatment with cisplatin, which is this first treatment here, the CTCs first had an initial spike, and then they went down, indicating that the treatment was working very well, to a minimum of about 9 CTCs per milliliter of blood. So they had a very great response as far as CTC numbers, and also clinically, the patient, you could tell, was feeling a lot better, and ECOG score was much better as well. At a certain time in her treatment, the treatment became toxic, and they had to take the treatment off of the patient. They did another treatment with cisplatin, with crizotinib. Here is treatment number 2, but the patient did not respond very well to the treatment. The CTC levels started increasing again, and the patient was just feeling a lot worse. Finally, they were put on a treatment on eribulin, which is the 3rd treatment, and again, the CTC levels went way down. And the patient had a very good clinical response to this 3rd treatment. So after we had finished this course, we went back, in fact, about a year later, and went back to the slides that we had in the freezer. And at these 6 different time points, we picked 5 individual cells and then did whole exome sequencing. And what we found is very interesting, that at the points where there were more CTCs in the patient, the CTCs were actually a lot more clonal. There was a smaller diversity in the number of variants that was found. However, when there was a good response to the treatment and the CTC total numbers went down, now you could see that the CTCs that we picked were a lot more heterogeneous. So it was an interesting finding that we had. And then in the right graph is another triple negative breast cancer patient. And again, the CTC number diminished after treatment with cisplatin. There was the first treatment. And then the CTC number increased again while they were on veliparib. And this patient actually had sequencing of their tissue sample, and it was found that they had an FGFR mutation. So at around day 190, the patient was put on ponatinib, which actually causes CTC numbers to drop to basically undetectable for a few weeks. And then towards the end of treatment, they started going up again. So after this, we also went back to the CTCs and sequenced 3 CTCs from the time point here at around 190 days. And we found that 2/3 of the CTCs were showing that mutation in FGFR that we knew was present in the patient. However, when the CTC numbers came back up after treatment with ponatinib, and I'll just remind you here, ponatinib is known to act against this particular FGFR mutation. After treatment with ponatinib, now we could not find any CTCs that had the mutation. So what you can see here is that the ponatinib actually eliminated the population of cells that had that mutation, and a new clone came up and was now the main clone that was in the patient. So you can see here that it's the power of liquid biopsy to measure real-time evolution of tumors at the single cell level. It would obviously be very difficult to do this taking continual biopsies of the tissue of the patient. So it really gives you a window into what's happening to the patient in real time. Then in this next slide, I'm showing a study that was performed with Minetta Liu at the Mayo Clinic, where we compared CTCs to cell-free DNA, and we did molecular analysis of both of these biomarkers from the same tube of blood. And we used the RareCyte blood collection tube that Leighton showed on his presentation. And then we compared the RareCyte blood collection tube versus a Streck cell-free DNA blood collection tube, which is kind of the standard that's used for cell-free DNA analysis. So on the left, I'm showing the comparison of the variant allele frequencies that was found between the cell-free DNA on the y-axis and the CTCs on the x-axis. And what you can see is more than half of the mutations were actually found by both of the compartments. However, 5 of the mutations were only found by CTCs and were not found in any of the plasma. And then 3 out of the 18 were only found with the plasma and not on CTCs. So you can see here that they're both cell-free DNA and CTCs are complementary. They both find most of the same mutations, and some of them, either CTCs or plasma, can find unique ones. But in general, this has been our results that we typically find more CTCs in the CTC. We find more mutations in the CTCs than we do in the plasma. And also, we think part of this reason is because with the CTCs, you have a more pure population of the cancer cells, whereas the plasma, you still really have to do a deep dive in the sequencing depth to really find the circulating tumor DNA. And here on the left, I'm showing the identity of the mutations that were found in the graph on the right, and then their frequency. And for the CTCs, the cell-free DNA either from the RareCyte plasma or from the Streck plasma. And the main takeaway here is that the CTCs and the cell-free DNA are complementary sources of molecular information, and some show actionable mutations that are were only detectable in the CTCs. And now another analysis that we did with the same data was when we look at the plasma now between the RareCyte blood collection tube and the Streck blood collection tube, there is near perfect concordance in the variant allele frequencies between both of these. So essentially with one tube of blood, the RareCyte tube of blood, you can get both the cellular compartment, or the CTCs, and the plasma compartment, which in clinical trials, sometimes it's difficult to get large amounts of blood, so it can be very useful to have just one tube of blood to collect both. In the following section, I'm going to describe in a little more detail the results that we obtained when we developed a HER2-ER CTC assay, and then we applied it to breast cancer patients. So the first step when we optimize a new assay is to identify cell lines that have varying expression of the biomarkers of interest. In this case, HER2 and ER are the biomarkers that we want to test, and to be able to quantitatively distinguish them by the level of protein that's detected on the CTCs. We pick the BT474 cell line here at the top, and it's the highest expression for HER2 and also positive for ER. Then our negative control line is the 468 cell line, which is negative for both of these biomarkers. And then the MDA, MB453, and OVCAR3 cell lines have intermediate expression of HER2 and negative expression for ER. So with these 4 cell lines as kind of our gold standard truth, we can now optimize the assay and then apply that assay to the cell lines to detect differences in biomarker expression. So what can be seen here in a graph and quantitative form is the same information that I just showed you in the previous slide with imaging data. And here what we did is each one of these boxes is 5 replicate slides that were run on a different day. And then we did 3 different runs on 3 different days with all of the cell lines. And so again, you can see here the interquartile range, the 10 and the 90th percentiles. And you can see here that the 4 different cell lines cluster very closely to each other because all the cells within a cell line have very homogeneous expression of the HER2 biomarker. And then you can see the differences between the high BT474s and then the negative 468s. So if we now use the 468s to test the specificity of the assay, we find over 99% of the cells were called negative of this cell line. And now sensitivity, you can get sensitivity of the 3 positive cell lines, where the high, the BT474 sensitivity is 100%, and then a little bit lower 453 cells are still over 99% sensitivity. And for accuracy, which is a combination of sensitivity and specificity, for the 2 high lines, we are again nearly 100% accurate. And then even with the OVCAR cell line, which is barely above this threshold for positivity, we still have an accuracy of nearly 90%. And this threshold of positivity is established with the cell lines that we've used as our gold standard. And basically, it can give you either a binary positive-negative result, which is completely objective and just based on the mean fluorescence intensity, or as I'm going to show you in the next few slides, you can get the single cell mean fluorescence intensity for each one, which is a quantitative number. After we have made the assay, we then test it on a number of clinical samples to show that we can detect it, not only CTCs, but the biomarker expression in a particular indication. So in this case, we tested breast cancer samples. And what you can see here, I'm showing just 4 CTCs from different patients. You can see the heterogeneity in the cells as far as the expression of cytokeratin and EpCAM, the size, the shape, the nuclear morphology, and all of these variables go into our algorithm that finds the CTC of interest in amongst the millions of white blood cells on the slide. And so you can see on the right-hand side is the HER2 expression in this column here for the 4 cells of interest, and then the ER expression. So with our assays, you can either have a binary positive or negative value, or the actual mean fluorescence intensity values that will give you the quantitation of that biomarker for that specific cell, and also the localization of that biomarker in the cell. So in the next section, I'm going to talk about a different assay that we made for the androgen receptor variant 7 and synaptophysin, specifically for prostate cancer cells. So as a quick background, the androgen receptor variant 7 has been heavily studied as a predictive marker for the use of 2nd-generation anti-androgen treatments, specifically in prostate cancer. And when you have expression of this marker on prostate cells, it's an indicator that the use of these treatments is not going to be effective and other treatments should be prescribed for the patient. Synaptophysin is a neuroendocrine marker, and it indicates that the epithelial cells have undergone neuroendocrine differentiation. And it's also a negative prognostic marker for prostate cancer. And both of these biomarkers can provide important clinical information about the tumor via the liquid biopsy. And also importantly, neither of these biomarkers at the protein level could be found in plasma cell-free DNA. Only with the proteins can you find expression. And so you can see here, after we went through full analytical validation of this assay, we then ran it with prostate cancer samples, and we could validate that we could find CTCs and enumerate them, and also quantify the expression of these two prostate cancer biomarkers. Again, we set off positivity cutoffs for both of the biomarkers, so you can determine just positive or negative threshold, or you can also take the single cell MFIs, and also you have the expression level on the cell, where it is in the cell of interest. The assay was then transferred to collaborators at the University of Southern California in the laboratory of Professor Amir Goldkorn. And he ran a clinical trial again in prostate cancer patients where they were, he collected 23 metastatic castrate-resistant prostate samples, and he did also a genomic comparison between CTCs and cell-free DNA and found much of the same results as we did in our lab. So the first thing he did, he also had cell search assay in his lab. So he compared on these 23 patient samples the levels of CTCs between the RareCyte platform on the y-axis and the cell search platform on the x-axis. And again, you find a very high correlation between the two assays or the two platforms, where there's a 20% higher sensitivity for RareCyte. So we found about 20% more CTCs on average than the cell search assay. And then on the right, I'm showing the dual biomarker expression on each one of the 517 CTCs that were found among the 12 positive samples in the study. And so you can tell here are the synaptophysin single positive cells in the top left, the ARV7 positive cells on the bottom right, and the double negatives. Interestingly, in this cohort, no double positives were found, which might be interesting biologically. Furthermore, then they did the single cell picking and sequencing. They found the germline variants, comparing the mutations found in the CTCs and cell-free DNA, and then in the white blood cells. And then they could find, by taking away the germline variants, they were able to find the somatic variants. Once again, similar to what we found, they found more mutations in the CTCs than the cell-free DNA. And because when you pick the single CTCs, you get a more pure population than with the cell-free, the variant allele frequency in all of these cases was above 20%. Whereas for the cell-free DNA, the variant allele frequency varied from a low of 0.3% to 63%. And then also some mutations were found on both the cell-free and the CTC compartments. Another analysis that they did was copy number variation. And again, they found copy number variability both in the CTCs and in the cell-free DNA and in both. And the copy number variation was found on the genes that you would expect, AR, synaptophysin, FGFR2, P53. So again, it just shows more applications that you can do with our platform. And then finally, I wanted to end the presentation showing some exciting unpublished data with the prognostic value of CTCs of our platform. So others have published on the prognostic applications of CTCs, for example, cell search in prostate, breast, and colon. What we did here is we had a small clinical study with Drs. Laura Kennedy at Vanderbilt University and V.K. Gotti at the University of Illinois. And they performed this on 28 patients with solid tumors who were enrolled in a checkpoint inhibitor clinical trial. So the tumors, as you can see here, were from a variety of different cancers, including most of them were lung, breast, prostate, and urothelial cancer. And even with such a diverse set of patients and a relatively small number of cases, we were able to detect a statistically significant difference in progression-free survival, which was dependent on the level of CTCs found in the patient's blood that was collected at baseline. There were 3 different collections, and then this is the data from the baseline collection. So it's very encouraging data, and we're putting it together for a publication. And what it confirms is what we've observed in many of our other studies that we've performed in-house. The CTC number and basically how it changes over time is a very good indicator of the patient's response to treatment or resistance to the treatment. In this last slide, I'm just going to show an image of the assay that was run for the PD-L1 ERF1 checkpoint trial, and it was a dual biomarker assay. As you all know, PD-L1 is an immune checkpoint protein. ERF1 might be a newer protein for many of you. It's a transcription factor that actually functions upstream of PD-L1, and it regulates PD-L1 expression. So you can see here the PD-L1 expression in red, the nuclear ERF1 expression in green, and then CK-EpCAM in the merged channel, and then the CD45. So when this publication comes out, we can let you guys all know if you want to receive it. Send an email to info@rarecyte.com and we can put you on the list. So in conclusion, I just want to end by saying that RareCyte does have a variety of products and services for liquid and tissue biopsy that are available. And it spans everywhere from discovery research all the way through clinical trials. And the key advantages of our platform is the ability to develop multiplex assays and then sequencing single cells or tissue microregions. So we have a comprehensive, flexible, and reproducible technology that's available to assist you in your research goals. So this is my last slide. As Leighton mentioned, this webinar is a first in a series that we're going to have coming up later in the year. So be on the lookout on your email for announcements for the next seminars. And with that, I'll open up the floor to any questions that Leighton and I would be happy to answer. And thank you very much for your attention today.
Great, thanks, Arturo. Very nice presentation. I've been answering some questions as they've been coming in throughout your presentation, but we have a number of others online that I think might be good discussion. So I'll kick off with this first one. Does your assay work only with fresh blood or with frozen PBMC as well?
That's a good question. Typically, most of what we do is with fresh blood. As Leighton mentioned, we have up to 72 hours from the time of blood collection through processing onto slides. But we have had projects where we've had frozen buffy coat, either from phycol preps or red blood cell lysis or different methods. And you can actually put those onto slides and then do the rest of our procedures on the automated stainers, yeah.
Yeah, and then from the same attendee, have you compared fraction of cells seen with your isolation versus cell types from standard phycol buffy coat preparation?
Yeah, we did that years ago. In general, what we found is the phycol typically lost about 50% of the CTCs. And not only that, but it was very variable from tube to tube and day to day. So, we found a much higher sensitivity with our system.
And then I'm going to keep your focus on CTCs, Arturo. Are the CTC biomarkers detected and quantified by fluorescent second antibodies?
Yes. So it's all based on a primary antibody, and then there's a secondary amplification scheme that's put together on top of that primary. And it amplifies the signal significantly, hundreds of thousands of times, so that you can get expression of very low abundance biomarkers. And it's all fluorescent, so it's scanned on the instrument digitally, and it has, as I said, up to five channels for testing those biomarkers.
Okay, and then we have a question then that says, can the assay be used in Orion and perform the similar test? Maybe I'll take that one. So we've obviously focused on CTC analysis today. Orion is one of our latest technologies, which is at the moment focused on tissue. It enables up to 21 biomarkers on a single tissue sample to be imaged in a couple of hours, basically. And while that's an imaging technology also, we're focused on tissue in the short term, but we do see application to CTCs in the future. Obviously, Arturo has shown you some multi-biomarker assays, which is unique in the marketplace right now. But for certain indications like sarcoma, for example, where you might want to have a dozen different biomarkers, then Orion's capability will come into play there because we can do so many more biomarkers at the same time with that capability.
And I will mention, Leighton, that we are about to release a little bit later this year a third biomarker channel, which I didn't mention. But yeah, so very soon we're going to have up to 3 extra biomarkers available on the biomarker assay.
And then there's a question around what is the volume input of samples that you can process. I'm not sure if that relates to the volume of blood, which is 7.5 mils, or the volume in terms of throughput. We have multiple systems. One is a 2-slide capacity, more of a research-centered unit. And then the other one, a high throughput device, is an 80-slide unit. So for 10 samples at a time in an automated workflow. And again, the throughput is typically sort of 10, 12 minutes for imaging, and then another 8 minutes for analysis after.
Yeah, and as far as the processing workflow for the blood, one person can run up to 16 samples at a time. To run a single sample takes about 1 hour and 15 minutes from blood to slides, and most of it is hands-off time, just a couple of centrifugation steps. And if you're now going to go from 1 sample to 16 samples, it probably takes on the order of 3 hours to process from blood to slides. And it's all very easy to use. It's all just pipetting, minimal hands-on time, and again, very reproducible from day to day and lab to lab.
And here's a great question for you, Arturo. I know you get asked this one quite a lot. Are the system thresholds defined by the operator or by RareCyte?
So that's a very good question. So the thresholds are set by the investigator and the particular biomarker, and it really depends on the positive and negative cell lines you've chosen. So that's where it becomes really important to choose the correct cells to give you that differentiation between the positive and the negative. But these are, our system is completely open so that we allow the users to set their own thresholds. And then obviously you'd want to test those thresholds against clinical samples because they can be different than the cell lines.
Yeah, and then related, Arturo, and you talked about this a little bit in the presentation, but maybe just build a little bit more on your last answer. How do you determine sufficient positivity in terms of MFI for a given biomarker?
Well, that's a good question. So one thing that's important when we do the assay development is not only how well the two, what the mean fluorescence intensity is of the different cell lines, but also how separated they are from each other. So certain cell lines can have very homogeneous expression, very, very tight. Those box plots can be very tight and then very separated. Whereas for other biomarkers, for example, biomarkers of cells that are cycling, you can have a very diverse biomarker expression within a cell line. And in that case, then you're going to not have as much of a separation between the positive and negative. So it really varies on the biomarker of interest, the cell lines you've chosen, of how well those two populations can differentiate with a single cutoff. For example, for Ki-67, if you use cell lines that are spiked in, they're growing in the lab, you're never going to find a completely positive or a completely negative cell line. So there's some tricks you'd have to do in that case to determine the positivity threshold. But yeah, each biomarker is going to have its particular things that you need to find out when you're doing the development and then be able to differentiate the positive from the negative.
Great, and then I think we just have a couple more. What was the read depth for the VAF calculations? Oh,
so that was done by the USC team I don't remember what the read depth was. Yeah, that's a really good question, and we have all of the names of the people that are putting the questions in here. So I think we'll get back to you on the particulars of that via email in the next day or two.
Great, thank you. And then maybe we have time for just one more. Could you share a CNV profile of selected cells? And how much is the distribution different from MDA in terms of dispersion of the distribution?
Yeah, so the MDA method to do whole genome amplification, we used to use that a lot. We actually moved away from doing whole genome amplification before single cell sequencing. The reason for that is that what we found is if you go directly into library prep from the single cells, your false positives, false negatives, and your coverage. So your false positives, false negatives go way down, and your coverage goes way up. So what we do now in general is not whole exome, whole genome sequencing, although it can be done and it works very well. What we do now is more targeted sequencing with panels, and those panels are for the 60 or so most highly mutated genes in solid biopsy, in solid tumors. And so you have about 600 or so amplicons, and you really get really good sequencing depth and very few false positives and negatives that way. So we've kind of moved away from the MDA method of amplification. But again, it depends on kind of what the goals are. Certainly for more I guess discovery biology, you might want to do whole exome or whole genome sequencing.
Terrific, thank you, Arturo. So with that, we're right at our time. We'll end the webinar today. Thank you for attendance. Thank you for your great questions. I hope you found this information useful, and feel free to reach out to us if you have any more questions or would like further information. Thanks so much and have a great rest of the day.